black, crush 'em like crazy.
Welcome to this lovely big room that we have all to ourselves for this session. Our thematic topic, the role of the host-associated microbiome in shaping life histories. And when we kind of proposed this session, we realized actually that's quite a big topic because when you talk about microbes that can be fungi, bacteria, and viruses, when you talk about the host they're associated with, that could be, of course, anything from the plant or animal kingdom. And then the way that things can shape life histories, of course, span the full continuum from pathogens to mutualists. And we're very lucky to have as our keynote speaker Dr. Thorin Helgeson, Senior Lecturer from the University of York, who has worked on a number of host microbe systems, who is going to be introducing the session as our keynote speaker, talking about hosts and their microbiomes, a question of scale and diversity. Thorin.
Thank you very much, Zav. Let me just put that on. Right, okay, that's me. Good morning. You're more than welcome to sleep through this. I accept it was a party night last night. It's a privilege to be opening this session today, particularly when it's hosted by Zav, who, when he left York University, was very confirmed bird person, and it's a real delight to see that we've drawn him back into the dark side of microbiomes and parasites. What I'm going to talk about in this slightly longer talk to begin with is part personal journey and a bit of science as well. I'm going to talk a little bit about how the discipline has changed or evolved in the last 10 or 20 years, and possibly how that affects the experiments that we do and how we do the science we do, with a bit of a personal journey in there. And then I'm going to give you a couple of examples going on in my group at the moment, and then perhaps make some suggestions about where we go from here. It's very clear from the program that we've got for the rest of the morning that there's a huge diversity of systems and questions and approaches being taken to microbiome research, and this is something that we need to look at as we're going forward. So, this is the pointer. Oh, I've got a pointer. This is a graph that I scraped from Web of Science at the weekend of all the papers that mention in the title or abstract the word microbiome since about 2000 onwards. Handful of mentions in the early 2000s at the turn of the century. How weird does it feel to be saying that about the 2000s? To me, anyway. And then we start to see this increase in papers coming up in 2008. If we look at that point there, that's when publications start to come into the system from 454 sequencing. We see another increase in this sort of exponent around about this point, and that's when we start to see high throughput Illumina sequence gain more traction across disciplines and moving out. What this suggests, microbiomes by definition microbiome research by definition produces big data sets, and that means that generating the data doesn't appear to be the limiting factor anymore. I certainly know that from my work, and I'm sure that's a common experience in the room. So we need to think quite carefully about how we progress the science that we do in the light of this change in how it works. So what I wanted to sort of briefly go through is how these things progress and how it works. We always begin with our burning question. The complexity of the questions and the talks that follow this is really fascinating, and how much we've been able to utilize these methodologies to answer interesting and exciting questions in ecology and biology. Then we move to it's mostly omics, to be fair, not exclusively, but mostly. What technology we use to generate that data? What target are we going to use? What platform are we going to use to do it? And then, of course, it's the dread question of analysis. There's an awful lot of us in my group who are mostly here in this room, and there are an awful lot of people, I'm sure, in this room and elsewhere who've had to learn an awful lot of Python and R code in the last 5 years. So the questions then arise. This has become a big and complex subject with lots of different complexities and different disciplines that each have their own legacy within a single project. The question then arises, which of these components should drive the other two? Should you be an expert in all three, as an individual project person or a group? Can you be an expert in all three? This is not a trivial question. Now, since it is Christmas, and this is not a pantomime, but nevertheless, I thought we'd have some audience participation. What I want you to think about is where on this spectrum you as an individual lie. It's a little bit, if I put my hand up, then I can see you all like that. Where on this spectrum do you lie, and where do you see your primary expertise, all right? So I'm going to ask you, if you see yourself primarily as an ecologist or a biologist, raise your hand now. Okay, that's a goodly portion of the room. Are you a chemist, biochemist, molecular biologist, somebody like that? One hand, I don't know who that is. And finally, are you an informatician? Do you see yourself primarily as a coder or a computer person? One, two, couple of hands there. Oh, look at that, you see. I made a prediction, which I think is borne out, that we'd be mostly ecologists with a couple of bioinformaticians, and the fewest representation would be for biology and the chemistry, for the biological chemistry. Yet, as we see from the original graph that I put up, our publications suggest that we're very heavily driven by the technology available. I know that's been true of my career, and how as new things come onto the block, you try and utilize what's available to you. But what does this mean for the science that we do, and how we choose what we do, and how we implement it? So this is the personal journey slide. This is my rant slide, right? This graph here represents through time. These points each represent data of mine or a data set that's in progress. That's the data set in progress. This paper is not mine because I'm not that old. It's Luc Simon's paper in Nature in 1992 that had the first arbuscular mycorrhizal sequences published in it, and they only had 8 sequences in that paper, and it got into Nature. Defy you to do that now. And what we can see here is the number of sequences put to get a decent paper, the number of sequences that you need to gain that traction with publications has increased massively. And it's a bit like Moore's law in that it has a doubling time of about 2 years. Note the scale on here. That's logarithmic. And what we can see here is that as the really high throughput next generation sequencing systems have come in, that even that is going exponential. So the amount of data and the amount of work we have to do to get data has increased exponentially through my career. I arrived at York in 1996, the same month that we got our first ABI 377 fluorescent sequencer. So I've never had to do sequencing with isotopes, so I feel I dodged a bullet there. But I've been, in this space of 20 years, I've been able to develop my science as the technology comes and goes. And the privilege that that gives you is that you get a bit blasé about the next new thing coming along because you see things come and go. I've seen fluorescent Sanger sequencing come and go. I've seen 454 come onto the market and basically cease to be supported by its manufacturers less than 10 years later. Illumina is the big thing now, but there are other things on the pipeline. But because I can sort of grow and develop with it, it's been easier for my generation of scientists, I think, to do that. But for early career scientists coming in at this stage here, you're being thrown into an incredibly deep pond in a completely different landscape. And as I have no doubt that the talks that follow me will demonstrate, the discipline's done an amazing job of integrating these technologies into ecological sciences. But the question then becomes, how do we, as a community, help early career scientists moving into this very complex field develop the skills and flexibility to be able to future cast and embrace the next thing when it comes along? And this has been ongoing in my group. We've done a lot of my sequencing in my group in the last 12 months, and it's been really interesting watching people new to this come and take on this technology and fight with it to varying degrees. So I think as a community, this is something that we need to embrace. How do we help early career scientists judge for themselves where they sit on this spectrum and which expertises they need to develop? And my plea would be that the challenge for me has always not to be to lose sight of the question. What data do we need to answer the question? and not fall foul of the what data can we get. What's that one? So with that in mind, what questions, now the science, that's the rant over. Now the science, what kinds of questions can microbiome research feasibly address? I'm going to give you 2 short case studies at completely different scales, just to sort of illustrate the breadth of what's possible with this kind of technology and the different kinds of questions you can answer. Honeybee gut microbiomes, which I know are coming up later in the session, and soil fungi and tropical forests as well. It will cover a range of techniques from traditional Sanger sequencing, TRFLP and Illumina-MiSeq. I won't go into the details of that. I'm sure most of the audience know them already. But keep in mind what I've said about embracing new technologies. First up, we have Foulbroods. This is a honeybee, that's a honeycomb hive. The Foulbrood diseases are bacterial diseases of honeybee larvae. The Foulbrood, American Foulbrood, is called by a firmicute bacteria called Painobacillus larvae. It's a notifiable disease in the UK, and the only cure for this disease is to burn the hive. And that's because the larvae in the cells in the comb get infected by the bacteria. The bacteria eats it from the inside. The larva dies, and the diagnostic test for this disease is particularly horrible because you stick a matchstick into the cell and you pull it out into the sticky rope, so it's called the sticky rope test, which is particularly grim. Healthy larvae like these pupate and the cell is capped. The dead larvae that are full of spores of the bacteria are cleaned out by the worker bees, who then proceed to spread it all over the hive. So it's a pernicious disease, and really the only solution is to just kill them. So it's a problem. A student of mine who just finished this year, in collaboration with the British Beekeepers Association and with FERA, Food and Environment Research Agency, decided to, we had a project where we were looking at the epidemiology of this disease, and the first aim of the project was to identify ways of identifying individual isolates of the disease, so the within species variability. The aim of this was so that the bee disease detectors, the bee disease unit, could detect whether an outbreak of the disease was a reoccurrence of an old strain, of the existing strain, or an invasion of a new one. So we needed a system that could separate out the different varieties. This is an MLST, it's based on straight Sanger sequencing. You get a number of genes, there are a number of polymorphisms within those genes. You count up those polymorphisms, and this is the relationship between the different sequence types that emerge. The distance between them on this diagram shows how far away, how many differences, gene differences or base pair differences there are between them. Painobacillus divides up into these 3 groups called eric types. But that's a very, very tiny proportion of the genome. So what she then followed up with was a comparative genomics approach of nearly 30 different isolates. We got the whole genome sequence, stripped out all the unshared genes, and aligned them all and looked at the relationships between the genomes. Right, it's a slightly complicated diagram. Focus on the top left as you're looking at it now. This is the whole bit. If we do that, we can see that the eric types are preserved. So these are all the blue ones, these are all the green ones, and the more distant group, the eric 4s and 5s, are still preserved. So the good news is that the whole genome sequencing reflects what we know from various phenotypic and gene marker studies. If we home and expand the blue bit here, we see a slightly more complex and interesting picture, which is something that we're now trying to follow up. If we focus in on this group here in the yellow, there's 3 or 4 types in there, 3 or 4 individual isolates that are in there. This yellow group here includes this sequence type, this one, and this one. So the genome similarity, the genome level similarity, is not congruent with the relationships that the MLST scheme implies. So while the MLST can still track differences between isolates, what that means in terms of virulence, behavior, spread, growth, and all of those other genetic characteristics doesn't necessarily hold. This is further complicated by the fact that this particular MLST type is also represented in that group there. So there's a lot of work to do now looking at virulence and diversity within the species. This isn't microbiomes, I hear you think. This is just a pathogen. Well, the reason why this is interesting in a microbiome context is because Foulbroods are gut diseases, and they interact with the gut microbiome. There's some evidence which suggests that disease progression and things like that depends on what the gut microbiome is. And an earlier study using just a straightforward TRFLP approach where you're looking for differences showed firstly that different stages of the honeybee have completely different gut microbiomes. The larvae and the pupae start off down here, and the workers, as soon as they emerge, and they pupate and emerge and are exposed to the environment and the other workers in the hive, they develop a completely different gut microbiome, which is interesting. And what we also see are species differences. In this graph here, this is a study that was done on Thai hives. And the student who came to York to do this brought with him samples from 2 different species. So Apis cerana is the Southeast Asian honeybee, which is native, and it's only semi-domesticated in Thailand. They use natural hives for it, whereas the European honeybee is managed in hives, and it's often supplementary fed. And what we can see here is that the 2 different species, even where they're co-located in space, so that the hives will be in the same location, they have completely different gut microbiomes. And in the paper, we attributed this to different diets and different foraging strategies done by the 2 species. Now, you put that in the context of exposure to pathogens in the gut, and then we can start to see how that might make a difference, and it might make a difference to bee survival. And colony collapse disorder and all of these things are really important with protecting honeybees and conserving pollinators. So this is work that we're now following up in a subsequent project. So perhaps we'll see more about this at BES next year. Completely at the other end of the scale, still in Southeast Asia, but at the hectare scale now. The other project that we've got going on in the group at the moment is based in Danam in Sabah, Malaysia, which is up at the top there. This is the bridge that goes into the 50-hectare plot that's monitored in the Danam Valley Conservation Area. That's not me on the bridge. I have a photograph of me on the bridge. It's a very, very high bridge. It's not for the leery, that one. What we're looking at in this project is a sort of trophic level response and the interaction between host diversity, between the fungal associates and the microbiota in the soil, and how that microbiota processes different forms of phosphate in the soil. The context for this is that most tropical soils are P-limited because they're old, weathered soils, and there's not a huge input of organic matter because it recycles so quickly. So what we're trying to do is to work out whether the distribution of different types of phosphate indirectly mediated by phosphate use by microbes then determines the spatial diversity and community structure of the tropical forests themselves. I'm now going to present you the hot off the press first pass at fungal diversity that we've got from the site. We got the data off the MySeq about 3 weeks ago. It's been chunted through the bioinformatics, and I drew the graphs at the weekend. So this is, I'll apologize now for it being a bit sketchy, but there's an awful lot of other metadata to go with this, so watch this space. So we basically asked which are the dominant fungal taxa. Is there a difference between the fungi present in the bulk soil, or is it compared to the fungi that are inside and around the roots? We had soil cores, 4 centimeter diameter soil cores within 1 hectare quadrats, lots of samples within each quadrat, and the quadrats themselves are about 300 meters apart. Separated into soil and roots, we did total fungal ITS sequence PCR, and these were next era barcoded and sequenced on the Illumina MySeq. That bit there, the contaminated kits, is there for anybody who's been doing MySeq in the last 6 months and has been fighting with Illumina for replacement kits on the 300 base pair kit. Ta-da! So proud of this graph. Just to highlight, that is 3.3 million reads. really does feel a bit like a hammer cracking a nut, this, because of these 3 million plus reads, the top 5 species that come out of the analysis account for 50% of the total. And I am sitting there looking at that spreadsheet thinking, did I really need 3 million reads to be told that half the half the community is only 5 species? But there you go. They're mostly saprotrophic fungi, some ascomycetes, some basidiomycetes. The literature suggests they are all of Southeast Asian origin, so I'm confident that that's a real result and not contaminant. One of them is a polyphenol degrading cryptococcus. So they're sort of complex degraders, just the kind of thing that you would expect in a tropical soil. This is a constrained ordination. What it suggests very strongly is that the main thing that discriminates these two plots is where they are, its spatial location. This polygon represents plot 1, and this polygon represents all the plots, all the samples from plot 2. So distance is going to be the key factor here. The different coloration shows the split between the root fungi and the soil fungi, and we can see there's a little bit more overlap there. So clearly there are some species that are, they're probably the things like the ectomycorrhizas and other taxa that sort of occur in both niches, and there are clearly going to be some which are restricted to one and not the other. So there's a greater degree of overlap here. This is the first tranche. Joe Taylor, my postdoc on the project, who is working on this, he's in Aberdeen as we speak, extracting the rest of the samples to do this on all of the rest of it. So again, watch this space. We may find more about this in the next coming year. Okay, so we come back to this. The examples that I've given you include techniques from across the spectrum, Sanger sequencing, TRFLP, Illumina, and indeed ion torrent sequencing as well. That's another technology that's kind of floating around in the background and everybody's forgotten about. The biggest decision for me as a scientist doing projects like this is that I realized fairly early on that I couldn't possibly do all of this. And gone are the days when you could pour your own polyacrylamide gels to load on an ABI 377 and get the sequences yourself. It's, you have to be an expert in too many areas, and we know how Britain feels about experts these days. So you limit your expertise. And the biggest decision for me in this has that I've decided to limit delegating the bioinformatics. That is a steep, steep learning curve for me because I'm not a natural coder. And, but for me, it's always been important to keep a control of the data so that I could see what the species are and actually relate that data back to the ecology so that you can iterate it and then ask the next question that's going to give you the most sort of fruitful hypotheses for further research. What I have decided, though, is that I do not do the middle bit. There are people who are much, much better at prepping the samples, running the samples. It's an economy of scale that we simply can't justify keeping in the lab, and we outsource all of our actual next-gen sequencing. That said, it's very important to understand what the systems do so that you can effectively quality control it and work with the providers to make sure you get the kind of data that you need. However, I'm not here to present solutions because the optimum solution space is very, very big. And I think the really interesting question to take away from this is, what other possible solutions are there? How do you approach this in your group? Are there different ways of doing this? Because I don't want to get stuck in a silo either, and I'd be quite interested to hear your comments on this when we're done. So just to conclude, these techniques are incredibly effective at identifying diversity, too effective sometimes, and the novel functions in the environment, taking that forward into who's doing what and where and why are they doing it. This is absolutely fundamental to traditional ecology, and we now have a window into the microbial box that we simply never had before. There are literally no limits, and I do overuse the word literally, but in this case there are literally no limits, because any biological process can in theory be studied. It's just a question of how you decide to use those platforms to do that. And again, the breadth of the talks that follow demonstrate that. But linking these complex community structures, these complex data sets, to host diversity and function, and this is what remains a challenge. And my final point here is that we all need to be future casting and looking ahead to what the next technologies are going to bring and whether we can incorporate that into our research, and if we want, and what can we do with it if we do. Higher throughput sequencing is on the way. PacBio is becoming long read sequencing, and really high throughput sequencing is coming on. Portability and field-based tests, that's my particular holy grail, and the one that I really, really want. Nanopore is the one to look for there. If you haven't come across nanopore sequencing before, I urge you to look at it now, because this is a paradigm shift, and it will very, very much change the way we do things. And yeah, work on that. I still have to work on my Python skills. Right. So this is my final slide. I hope I haven't run over time. And I'm going to leave you with my favorite John Muir quote. I always end talks with this quote, because it's, everything is bound fast by 1,000 invisible cords that can't be broken to everything else in the universe. The longer I stay in science and the more complex the projects I do become, this becomes truer and truer with every passing day. And as I say at the bottom there, it's as true of science disciplines now as it is of environments themselves. And I'll crib a comment that Duncan Cameron said to me a while ago, which is you have to learn how to collaborate your way out of trouble. And I think that's as good a place to end as any, with acknowledgements to the people who did the data, my various PIs and friends, and of course the funders. And thank you very much for your attention.
Thank you, Thorin, for that excellent overview of plants and pathogens and animals and beneficial microbes all in one. That was excellent. Is there time for some questions? Okay, we have time for some questions. If someone would like to ask one, just put your hand up and I'll run over to you.
Hi, Thorin. Thanks for that. It was really, really interesting talk. It's a bit of a philosophical question, so apologies if it's a bit of a difficult one.
It was a philosophical talk, to be fair. So,
something you were hinting towards at the end of your talk was sort of where the field's going in the future. What development or what ability to gather new data kind of excites you most? Be that a technological advance or bioinformatics or whatever.
I'm not a good enough bioinformatician to futurecast that, I do, I'm told. With technology, though, Nanopore is definitely the one to watch, because they're starting to ship all of their things at room temperature. They're aiming for field testing. And I heard a rumor that that's where they're going with it. And there are possibilities with that technology of taking things like min-ions and miniature versions of min-ions out into the field and doing your sequencing direct. And there's so many sort of caveats about sequence data. There's, oh, your things have been preserved wrongly, blah, blah, blah. If you can actually do all that straight away in the field and get your long-read sequencing in situ, that's a game changer as far as I'm concerned, particularly if you can do it for RNA.
Thank you.
Okay, in the interest of times, I think we have to leave it there. So
No
we thank Thorin once again for an excellent talk. And I'm just going to stand here rather than walk all the way around to the stand again. But continuing on the plant theme, we now have Dr. Davide Bulgarelli, who's going to be talking about tracing the domestication route of microbiota in barley.
Okay, great. Can you hear me? Yes. Fantastic. So, thanks for this kind introduction, and also I'm very glad to be here. Firstly, because I'm not an ecologist by training, so please keep in mind in the Q&A session this. And but I'm very glad to share with you our recent insights on the plant microbiome and how the plant microbiome can influence plant life. So basically, in the next 13 minutes, what I want to do is try to convince you that the plant microbiota rocks, and in particularly the microbiota that thrives at the root-soil interface. Then I will use a story that we are developing in our lab to explain how to study the plant microbiota and what kind of implication this could have also for translational biology. And at the very last, I will use a slide to basically illustrate where I think the field of plant microbiota interaction is going in the next years. Okay, so to begin with, why bother with the plant microbiota? Plants are autotrophic organisms, so they can use sunlight to fix carbon dioxide into organic compounds. However, to do so, plants need soil. So from agricultural point of view, the vast majority of production is soil-derived. And plants in soil face a myriad of microorganisms, and most of them can interact with plant and can affect plant both positively and negatively. So from plant biologist's point of view, understanding the interaction between plant and the microorganisms that thrives at the root-soil interface is absolutely crucial to unlock the potential of plants for agricultural production. And now, what we know of the microbes that thrive at the root-soil interface is that this is not just a random assembly from the surrounding soil biota. So there are processes ongoing of microbial selection, and those processes that progressively differentiate the soil biota into the plant microbiota are driven, at least in part, by the plant itself. And actually, this is the main question of our lab. So our plant genome can dial into a myriad of microbial genome, and what kind of implication this has for plant growth, development, and health. As I mentioned, I'm a plant biologist with a clear interest for potential application. And from agricultural point of view, I've realized when I was establishing my own group that perhaps there is another question that we should ask first, which is this one. So the life histories of plant has been somehow manipulated by humans since the Neolithic Revolution 10,000 years ago. So this selection profoundly modified the phenotype of the plant as eloquently represented here. This is an ancestor of the cultivated mice, and this is what we grow nowadays. So the question is, what if domestication impacted also on the capacity of plant or interacting with the microbes that are thriving at the root-soil interface? So therefore, I realized that to answer this question, I would have need a model that is a cultivated plant, such as barley. Barley is a worldwide crop. It's grown from Canadian prairies till Australia, passing to Scotland, where it's the backbone of the feedstock and malting and distilling industries. At the same time, is an experimentally tractable organism. So the genome is available. We can assess mutants, we can assess a population, we have resources to interrogate the barley genome, so therefore it's an ideal model to study the interaction between plants and microbes at the molecular level, and perhaps most importantly, at least for our lab, the wild ancestor of modern cultivated variety are still available for experimentation, are present in the area of domestication, and are interfertile with cultivated varieties, and therefore we can study plant microbiota interaction in a domestication framework, in a plant domestication framework. So, and today I'm going to tell you as I said, one of the stories that we have ongoing in the lab. So this picture displays one of the area of domestication of barley, which is the Fertile Crescent. In this area, we still have different wild barleys growing, and they are now organized in a way that reflect the area where they grow, such as the northern part of Israel, the Golan Heights, that has a very distinct pedoclimatic condition compared to the coastal plain, or the more central and desert area of this region. So basically, wild barley in this environment is facing a multitude of soil environment, and we wondered whether this capacity of wild barley in thriving in such a condition has somehow been, let's say, grasped by early farmers and used during domestication to make sure that barley could be grown in areas that are very different from a Mediterranean environment, like Israel, for instance, in Scotland and in Scottish soil. So what we have done is we have collected the seeds of this wild barley, and we decided to grow them in our own greenhouses. So this is the institute where the Division of Plant Science at the University of Dundee is based. The James Hutton Institute is basically on the outskirts of Dundee. This is the River Tay. So we got a lot of fields around here where barley is cultivated. So we decided to sample some of those fields, actually 2 different fields, we collect the soil, we transfer into a greenhouse compartment, so we grow them, those plants under controlled environmental conditions. We decided rather drastically to leave aside any other environmental parameter that could influence plant microbiota interaction and focus on the soil component. So what we do is we collect the plants, both wild barley and domesticated barley, once growing in the greenhouses. We collect the so-called rhizosphere, so the thin layer of soil surrounding the roots. And as a comparison, we have also some pots where there are no plants. This is what we consider bulk soil. And what we do is fairly simple. So we collect these, we shuffle everything into a sequencing machine. We use a MySeq machine. And depending on the target of the sequencing, whether this is a phylogenetic marker, such as the 16S ribosomal RNA, or the totality of the DNA, we can address two questions. And invariably, as eloquently demonstrated in the previous talk, addressing and answering this question rely on an extensive analysis from bioinformatic point of view. And in our group, we are strongly committed with open science, so we use open source tools such as R and many other, because we realize that this is a strategic decision, and this will allow us basically to share data and protocols with other labs. So basically, these are some of the results that we gathered. So this is a census, it's a bacterial census of the barley microbiota in the two soil that we have used. And as you can see, at least at first glance, there is a clear barley effect. So these are the three ecotypes. They have a range of several samples. These are domesticated one, and this is the implanted soil. You can see that, again, as I mentioned, there are processes of selection. Bacteria, such as Proteobacteria, Actinobacteria, and Bacteroidetes are enriched, and others, such as Actinobacteria, Acidobacteria, are depleted in the barley rhizosphere. But there is no clear effect, at least here, at the level of the individual plants or the different soil. And perhaps because we are very high in the taxonomy classification of the microbiota. So if we go down at the terminal node that we can address with our sequencing technology, the so-called operational taxonomical unit, so what we can start to see, for instance, using a constrained ordination, is a clear soil effect. So depending on the two soil type, depicted here by different shapes, we can separate our samples. And within the barley samples, we could see that members of the desert ecotype tend to cluster separately from the other. However, this is something that we can appreciate here, but we couldn't really quantify using this approach. So we decided to move on and use basically an enrichment analysis, identify the individual bacteria that were differentially enriched between desert and the other ecotypes. And what we have observed is that members of the northern coast ecotype and domesticated have the tendency to accumulate a certain number of OTUs that are not so abundant in the desert ecotype. This is the aggregate abundances of these enriched bacteria. And this pattern is somehow conserved in both soil, although the distribution of the individual taxa is not conserved, and their abundances is not conserved. However, there is a conservation across soil type, and this is predominantly manifested at the level of members of the phylum Actinobacteria, that is exemplified here by this Terrabacter and Phytococcus-related operational taxonomic unit. So what you can see here, moving from the implanted soil to the domesticated one, is a sort of progressive gradient where domesticated tends to enrich more these bacteria compared to member of the desert ecotype or the other wild barleys. So then the question that we ask, although these data are still preliminary, is why only Actinobacteria, or why predominantly Actinobacteria and not other members of the microbiota? And we are still trying to address this question. And what we have realized by looking at the published data of other species, in particular this Arabidopsis microbiota paper that was published last year, is that if you look at the functional diversity of these bacteria, so basically those guys have sequenced up to more than 400 individual genomes, and then they have compared the functional capacity and functional diversity of this genome, and they've observed that within the phylum Actinobacteria, there is a strong degree of conservation of functional capacity. So functional diversity goes side by side with taxonomy diversity. So that perhaps this is one of the reasons why Actinobacteria tend to be conserved even across soil types. And clearly what we don't know, whether this represents a sort of domestication syndrome, for barley, and perhaps most importantly, what they are doing in the barley microbiota, whether they are positive or negative, at the moment we still don't know. And this is actually my last slide, and is where I think the plant microbiota science is going. So we see, let's call it, we plant microbiota scientists, we see the future in the hands of microbiology rather than computational biology. So we have realized that, yeah, this is absolutely important. We need to know who is there. We need to know how they are somehow related or how they are influenced by different parameters, plant species, plant genotype, or soil type. But now we are in the position to isolate them in a rational way under, let's say, microbiological conditions. So basically, tease apart these invisible bonds that were mentioned before and try to reconstruct them using reductionistic approaches, because we strongly believe that this is the way to gain insights into the functional significance of those communities for plant growth, development, and health. And this is it. So if you have enjoyed my talk, I think that you should express your gratitude in particular to Rodrigo, the PhD student who spearheads this project. Conversely, if I haven't convinced you, I'm standing here and I'm happy to take any question. And of course, I would like to mention the funding bodies that are supporting our lab. And again, I would like to thank you for the attention.
Okay, we do have time for a quick question.
Hi, that was a really interesting talk. I'm a plant biologist as well, so I shouldn't be here either. It's nice to see some other people here. I understand that a lot of soil microbiota aren't actually culturable. And I do like the idea of making your own communities and seeing how those interact with your different lines, but do you think you might run the risk of missing things out that actually you can't culture?
Okay, so actually this is a very interesting choice of word. You say the soil microbes are unculturable, and in fact this is true, but the plant microbiota is just a fraction of the soil microbiota. There is a strong selection. You have up to 52 different phyla in soil, in terms of soil microbiota. Plants, an handful, maybe 5 of them represent the vast majority. This Nature paper that I mentioned before demonstrated that up to 65% of the Arabidopsis microbiota can be indeed cultured under laboratory conditions. And once reassembled using reductionistic approaches, revealed that the assembly is strongly similar to what you can find in soil. Clearly, yes, it's not 100%, I do appreciate this, but we are in the position to test at least different hypotheses to begin with, and maybe in the future we'll be in the position, with more genomes available, we'll be in the position to maybe identify why we cannot culture the other, let's say, 30%.
Okay, we're out of time, so please join me in thanking Davide once more for a great talk. Okay, we're going to stick with crops now, but paradoxically move into animal microbiota. So please welcome Chloe Hayes from the University of Liverpool, who's going to be talking about host-associated microbiota of Drosophila.
Hello, everyone. I hope you can hear me. My name's Chloe Hayes. I'm here from the University of Liverpool, and I'm going to talk to you today about the role of the gut microbiota on the behavior of the invasive crop pest Drosophila suzuki. So the formatting's gone a little bit awry there, but so this work I did for a couple of months at the University of Georgia in the United States. And in case you don't know where Georgia is, here's a lovely map. So it's one of the southern states of the US, and it's a very hot and humid climate, which enables it to have a perfect condition for growing a lot of fruit crops. So since the early 1900s, Georgia has been known as the peach state due to its massive production of peaches. However, since then, its blueberry production has surpassed its peach production, and it's now one of the biggest blueberry producers in the US, with an estimated total of $203 million blueberry industry just in Georgia alone. That doesn't account any of the other southern states that also produce blueberries. So with this massive increase in fruit production, there has arisen a crop pest called Drosophila suzuki. And it currently is accounting for 20% of an annual loss of Georgia's blueberry production, and that's just Georgia's, not any of the other states. So it's clearly quite a big problem. And we know that Suzuki was native to Asia in the 1930s, and then in the 1980s, a few populations arrived in Hawaii, and they stayed there for a little bit, and then about 20 years later, they'd reached the mainland USA in 2008, and then from there, they spread across the whole of the USA and even into most of Europe. So Drosophila suzuki is unlike any of the other Drosophilids, as they're able to feed on ripening rather than rotting fruit. And they can do this because the females possess a serrated ovipositor, which you can see enlarged in this bottom right-hand corner, which is it's quite a fierce-looking instrument, and it's got these sort of knife-like serrations all the way down it, which enables the female to saw through the ripening fruit and lay her egg inside. Then the resulting egg hatches, the larvae grows and develops on this ripening fruit, and then once the pupae, they pupate, and then the adults continue. So how have Drosophila suzuki made this ecological transition from rotting fruit, which is very protein rich, to ripening fruit, which is very protein poor? Protein is an essential nutrient in the growth and development of Drosophila, so it's quite a mystery how they've made this transition. And some studies have shown that adipokinetic hormone, or AKH, is linked to this. AKH is responsible for metabolism, for lipid metabolism and homeostasis, and is also involved in mobilizing energy from the fat body, food intake, and nutrient absorption in the midgut. And some studies have shown that AKH expression varies with protein content and starvation status. So on low protein contents, or when individuals are starved, they have significantly reduced AKH expression. So here we've got this beginning of a chain. We can see this diet change, which causes a change in AKH expression. And another study has also linked AKH expression to mediating pheromone expression. So how do they do this in Drosophila? Well, they do this through changing something called CHCs, which are cuticular hydrocarbons. These are heritable fatty acids that cover the surface of terrestrial arthropods, and blends of these hydrocarbons create an individual scent profile. And slight changes in these CHCs can cause massive changes in mating behaviors, as slight changes causes an individual scent profile to differ, which changes the way that individuals will react with each other. So this brings us to our big question. These other studies have only shown that diet changes causes this chain of effects, but they haven't actually ever separated the gut microbiota from the diet. So we're suggesting that it's actually the gut microbiota that's responsible for mediating this ecological transition. So this brings us to our study. We wanted to investigate the effect of the microbiota and AKH on essential courtship and mating behaviors. So to begin with, we asked, does it affect courtship behavior? Courtship is quite an important trait in Drosophilids, but especially in Drosophila suzuki. They have quite a complicated courtship dance compared to other species. And we also wanted to know if presence or absence of gut microbiota affected whether individuals were likely to mate or not. And then finally, we wanted to see if AKH was responsible for any of these changes. So how did we do this? Well, as I said before, Suzuki bury their eggs into ripening fruit. So here is a picture of eggs buried underneath this dietary media, which is why it's quite hard to tell what they are. So in order to harvest the eggs, you have to dig them out. So once we'd harvested eggs, they went through one of two pathways. The first was being washed in PBS and then placed onto a conventional diet, and conventional flies were reared. This was the first treatment. And then these eggs washed in PBS were also placed on the same conventional diet, but with the addition of extra microbiota. So this microbiota was harvested from stock population, the gut was removed, and a lysate created, and small amounts of this lysate was added to the dietary media. So they're the first two treatments. Eggs were either washed in PBS or washed in bleach. This is called egg decorionation, and it removes the outer layer of the egg, called the chorion, which contains most of the bacteria that the fly needs when it's developing. So we removed this, and then eggs were either placed into a completely sterile environment, so therefore flies that grew up hopefully had no microbiota present, or they were placed into the same sterile diet, but again with the addition of microbiota. So these treatments, adding the microbiota back, we were hoping to see that if conventional flies had acted one way with regards to mating and courtship, whether this axenic plus microbes caused them to act in the same way that the conventional flies would otherwise, or whether it would cause a different effect. So the first things that we looked at were mating and courtship behaviors, and we measured courtship effort and the proportion of mated pairs. And I've only got some preliminary data for you today, but this is courtship attempts, and so far we can't see any significant differences, but we can already see a trend beginning to emerge. These two conventional diets on the left have about double more courtship attempts than the two axenic treatments, regardless of whether microbiota was re-added back or not. And for the proportion of flies mated, we can see a pretty similar effect. So for the conventional treatments, they mated a lot more than the axenic treatments. And we can also see a significant difference between the conventional plus microbes treatments and the axenic plus microbes treatments. So clearly something is going on when we're trying to add this microbiota back. Something is going on there. So as I said, I've only got very preliminary results for you today, but that brings me on to what we're up to next. So we've already started to underpin changes in this link. We've got these mating behaviors at the bottom, and now we need to work our way back up to define these links to see how we got from this ecological transition to subsequent mating behaviors. So next we're going to measure the AKH expression and then see if we can correlate that with a change in pheromone expression. So we're going to do that using QRT-PCR, and we're going to measure the AKH and the AKH receptor expression, but also we're going to look at the immune gene expression to see if presence or absence of microbiota affects immune gene expression. For example, does having no microbiota cause a stress response in individuals, which causes a higher expression of these two immune genes? And for this, we're using dipterosin and metdecoin, which are standard measures in Drosophilids. But because we haven't quite finished these mating behavior measuring, we're also going to look at mating latency and copulation duration. So copulation duration is a standard test used for Drosophila mating assays, as it measures male investment into a particular mating. So Drosophila suzuki has actually now colonized the UK and is spreading throughout the country. So I've actually managed to get my own population here, so I'm going to carry on this experiment in this population, and then my lab that I was working with in the US is going to continue the work there, and then hopefully eventually we'll have these two results that we can compare and contrast against these two populations of Suzuki, as currently no one really knows how they've changed as they've moved across the world. And with that, I'd like to thank the lab that I joined at the University of Georgia, and also my supervisors here in the UK. Thanks very much.
Okay, we've got plenty of time for questions.
Yeah, I do have a question. So it's unclear to me, the microbiota that you had in your experiment, these has been collected from flies that are feeding on rotting fluid or fresh fluid?
On fresh fruit, yeah.
On fresh.
On the ripening fruit.
Okay, are you considering also as an additional control microbiota from other flies that are feeding on, let's say, rotting fruit?
Yeah, that could be an extra treatment to add in. Once we've examined these preliminary results, seeing how the microbiota changes from individuals on rotting fruit to ripening could be a good test.
Any other questions? Oh, yes.
Have you looked at whether your Azenic Plus treatment's actually working? Have you looked at the gut microbiota in those flies?
Yeah, we did actually plate out, just taking the guts out and plating it onto standard agar media to see whether anything grew. And for most of them, nothing did grow, but for some of them we had a few small cultures that we suspect is Lactobacillus brevis, because I've done some work on melanogaster here in the UK on that before, and it tends to be a bacteria that sort of, when you put the flies through this massive stress, then sort of more of those flare up than other ones. But yeah.
I have a question which is dear to my heart, and does that mean that British crops of fruit like blueberries are now under threat, and I might not be able to have them on my cereal in the morning?
Yeah, it does. So the
I knew it.
The population isn't that drastic yet, but it's already started spreading from the south of England up to the north, and it gets any soft-skinned fruit, so like blackberries, anything that grows naturally, they'll target. They're not picky, but we're doing what we can to stop it.
All right, I think we should move on there. Thank you very much to Chloe for an excellent talk. And our next speaker is Dr. Vincent Doublé from the University of Exeter, who will be talking about multiple pathogens in honeybees.
Good morning, everyone. So I will now talk about the bad microbes, and I would like to show you how important it is to take into account the multiplicity of pathogen infections in a host to understand the complexity of this response to pathogens. So eukaryotes live in multi-pathogen worlds with a diversity of threats such as fungi, bacteria, and multiple viruses that can infect a host. And to this multiplicity of threat of pathogens, there is also a diversity of response. This response can be of different aspects, could be behavioral, with the avoidance of diseased organisms, could be at the molecular level with gene expression, change in gene expression. I'm thinking about immune genes, for example. And also this response can be mediated by the microbiome and beneficial microorganisms. And so in my work, I'm studying this response to pathogens at these different aspects. And I will talk to you today about these two aspects, the molecular response and the response that is mediated by beneficial microorganisms. And the model I use in my work is the honeybee. That's eusocial insects, so they live in dense colony of thousands of individuals that share foods, that are in constant interactions. So this facilitates disease transmission. And in that sense, this model is really interesting. And in my work, so I'm doing experimental work in the lab. I've been using multiple pathogens, and in this work I will present today, these pathogens are gut microsporidia, which are nosema here, and also multiple RNA viruses. All right, so first I will talk to you about the molecular response of honeybees to multiple pathogens. And it started with a collaborative work that I co-organized in Germany to do a meta-analysis of 19 transcriptomes. Transcriptomes that were from 19 independent experimental infections, so where gene expression was recorded and compared between infected bees and control bees in experimental setup. So that come from recently published or soon to be published data. And here are the data. So we collected the differential expression of 7,000 genes across, so this is the 7,000 genes across 19 transcriptome, 19 experiments, and you can see the profile of the gene on this heat map. And you can see within these 19 transcriptomes of half of the data were obtained from bees that were infected with different Nosema species, the other half with viruses, and we also got some co-infected experiment there. So using this large data set, we were looking at the gene expression to multiple pathogens and test whether there is a specific and or a general response to these pathogens. And also we had a look to the evolutionary rate of differentially expressed genes. So first we had a look to the specific response, and it's known in other organisms that there is a specific response. So the response to viruses won't be the same as to bacteria, and there is indeed in the immune gene repertoire, there is specific pathways against different kinds of pathogens. And here, using these data sets, we were able to test, identify genes that were differentially expressed against fungi in this data set, but were not differentially expressed when there were no fungi in the experimental setup, right? So doing this, we did identify a specific response with antimicrobial peptide responding only to fungi, and also genes involved in apoptosis and autophagy, and it's known in other insect species that these mechanisms applies against Microsporidia. And we did the same against viruses, and we also found genes, and here I give an example of the immune genes from the Tollin AMD pathway that were also found in flies to have an antiviral impact. All right, but the other thing that we wanted to test, the other hypothesis we wanted to test using this data set, is there a general response against any kind of pathogens? So basically, it's looking at genes that were consistently regulated by any kind of pathogens, downregulated or upregulated. And yes, we found genes that were consistently regulated. We found here 160 genes that were significantly regulated, so 57 genes that were up there, so mostly red on this map, that were upregulated again in most of the data sets against different pathogens, and here downregulated. We got immune genes, and here it's interesting. We do have immune genes that can respond against fungi or against viruses. And we found that there is metabolic and catalytic activities that are downregulated after any kind of infection. So it's interesting here. We have a general response, which could be an interesting marker of infection, and also potentially, and I'm thinking about the upregulated genes, an alarm signal that could spread across the body to tell that there is an infection going on. And when we looked at the evolutionary rate of the genes involved in that general response, here you have in orange the genes that show increased expression with pathogens, and here decreased expression. We do find, when we look at the protein sequence of the honeybee genes and compare to other bee species, we found a faster evolution of the genes here. So potentially there is an impact of selection there on this general response. Then on the second part of my talk, I would like to also present a piece of work that went be done on the role of the gut microbiome in host response to pathogen in bees. So the gut microbiome of social bees and honeybees is rather simple and specialized, with few taxa represented. And the transmission is made by nestmates. So basically, when an adult is hatching in the hive, there is no gut microbiome, and after contacts with nestmates gets gut microbiome. So it's important to keep that in mind, because it actually means that we can manipulate it in the lab. And we know the function of this gut microbiome for digestion, detoxification, and defense against pathogens. So here I talk about one specific component, which is lactic acid bacteria, with a question here. So these lactic acid bacteria produce lactic acids. They form a film in the crop, which is at the beginning of the gut of the digestive tract of the bee. And they are used in medicine as probiotics. And we know that these bacteria have an antibacterial activity, and indeed they are used to control bacterial brood diseases. All right. So the question is, so we know this lab can have an antibacterial effect, but can lab protect against other diverse adult infections? So to respond to this question, we did a simple cage experiment with caged bees, and we gave the lactic acid bacteria or not. And then 2 days later, either we give a control solution or fungi or viruses. And we looked at the effect on different parameters. First, we looked at the survival. So this is a hazard ratio. It's a calculation of the probability of death of bees in the different treatments there. And we can see that when the lactic acid bacteria is present, there is a decrease a probability of mortality, so a highest survival when the bacteria is present in the control compared to the bees that didn't get this bacteria. And in the treatment with the pathogens, we also see an increased survival, especially with the fungi here. So it seems that bacteria is really important for survival, and even in the case of, and especially here in the case of infection with pathogens. But how does that work? So we also had a look to the dynamics of infection of these pathogens in these different treatments. And what we found is actually the two pathogens are doing better when the bacteria is there. So we had an increase in virus when the bacteria is present, and an increase in spore load of the fungi when the bacteria is present. So here we have clearly a case where we have a bee that is doing better with the bacteria, but also the pathogens are doing better. And it seems that there is a higher tolerance of these pathogens when the bacteria is there. So take-home message here, it's the importance of taking into account the diversity of pathogens to understand the complexity of host response. And by doing this meta-analysis, we do show that there is indeed a specific response to pathogens, but also there is a general gene expression response. And this is particularly interesting in terms of co-infection, and we know that co-infection can increase mortality in bees. And it's also important in terms of emergent diseases, and you're well aware about emergent diseases that that is problematic in the case of honeybees. And in the case of the three-ways interaction with the microbiome, we could see here that a bacteria that has a known antibacterial activity can also increase tolerance to other pathogens. And the idea now is to understand the mechanism behind that, because there is clearly not a direct interaction between the pathogens and the microbiome, or at least not with these bacteria, and to understand what is happening with the gene expression when the microbiome is there. So with that, I would like to thank many people for this meta-analysis. Many people participate to this work, but especially Yvonne Perschel and Christina Grotzinger and Robert Paxton. And for the experimental work, that was the work of Ted Desirotana-Watt in the lab of Robert Paxton. And thank you for your attention.
Questions, please. I will start with one. Because there's so many general immune genes upregulated in response to both pathogens, does that mean they cope quite well with co-infection, or do they still suffer quite a lot?
Well, in experimental work, we did show that co-infection between fungi and viruses increase severely mortality. Well, that was one experimental setup in terms of what's happening in the hive in nature. It's probably a more complicated picture, but yeah, potentially here we have candidate genes to understand what's happening in terms of interaction, in terms of synergistic interaction, which is seen in terms of mortality. So yeah, I guess that's something that will be investigated in the future. So this meta-analysis was really good to pin down a short list, core set of genes that have to be investigated more closely.
I'm going to hog another question if no one's going to be forthcoming. People get quite excited in the amphibian disease world about the idea of applying probiotic bacteria in the wild to mitigate infection risk. And I think you hinted at this, but is that something that people are doing with bees and these lab bacteria, or is it not feasible?
Well, obviously it's feasible to give them the bacteria as we did in the lab. So in terms of large-scale treatment, I'm not sure where it's going at the moment, but definitely, I mean, the bees are using it in the process of doing honey and also processing the pollen in the colony. So that's something to support if there is indeed a lack of this bacteria in a diseased colony, for example. So if there is a diagnostic like this, definitely that would be one answer to help to increase health in bees.
Okay, I think if there are no questions, then we'll move on. Join me in another round of applause, please. And our next speaker is Kieran Bates from Imperial College London and the Zoological Society of London, who's going to be talking about disease and host-associated bacteria in montane populations of amphibians.
Hiya. So about 10 years ago, researchers from Imperial College London and the Zoological Society identified a handful of populations in the Pyrenees, amphibian populations, which were declining for seemingly unexplained reasons. Analysis of some samples they brought back to the lab revealed that the driver of these declines was a then recently identified emerging fungal pathogen called Batrachochytrium dendrobatidis, or BD. Now, BD belongs to the phylum Chytridiomycota, and it has a global distribution, being found on every continent where amphibians are. It has a life cycle that's split into two key phases, a motile aquatic zoospore, which colonizes the host's skin, encysting and forming a sessile zoosporangium that produces further zoospores that are then released back into the aquatic environment where they can infect other hosts. The key pathologies associated with this disease are thickening of the skin and disruption of sodium-potassium ion pumps, which causes an ionic imbalance, eventually resulting in cardiac arrest and death. So my PhD specifically has been focusing on 5 populations in the Pyrenees, which have been surveyed every year since initial emergence. And what we found is that there's very diverging infection dynamics. So the populations which are in blue, we've described as having enzootic infection dynamics based on low infection intensity and prevalence and a resurgence in population abundance. And this seems to be consistent with the development of tolerance or resistance to BD. Conversely, some populations which are infected at the same time, shown in red on the map, have continued declines and have high susceptibility to BD. And we have the population abundance numbers for larvae in recent years, shown in this table here. And actually, in our lathe, the most susceptible population, we couldn't find any tadpoles in 2015. So this really is on the brink. Now, there are several factors which are likely to be driving these different dynamics, including the host biology, the pathogen biology, and indeed the abiotic environment. But I want to focus on the role of the skin microbiome. So there's a lot of evidence at the moment to suggest that the skin microbiome of amphibians may be important in the outcome of disease. So we know that certain bacteria isolated from the skin have antifungal properties and inhibit BD in vitro. And we also know that the microbiome is really important to overall host health, and a perturbation in the microbial community structure could have potential effects to the normal homeostasis of the amphibian skin, causing ill health. So in the summer of 2015, I went to the Pyrenees and I swabbed metamorphic common midwife toads from these 5 populations, taking skin swabs, which I then analysed using 16S metagenomic sequencing and to characterise the bacterial communities, and then qPCR to determine the infection load on the individuals. And unsurprisingly, the infection data shows that there's a high prevalence and a significantly higher infection intensity in the epizootic populations, which are in red. Now moving on to the bacterial data, interestingly, we show that there is significantly lower bacterial diversity on the populations that have the epizootic dynamics. And this could be down to a range of factors. It might be that the enzootic, the tolerant populations, have a naturally higher bacterial diversity, and this might be associated with increased production of antifungal metabolites, and also have a competitive advantage in terms of preventing the invasion success of BD. Alternatively, what we might be seeing is that in the epizootic populations, this reduction in alpha diversity is in response to the BD. So we know that BD causes skin sloughing, which is essentially where the skin comes away from the animal. And this has been shown in previous studies to have a negative effect on bacterial diversity. It also could be that the high infection intensity is weakening the host immune system, which is making it easier for opportunistic bacteria to then invade and become more dominant members of the communities. In terms of bacterial community structure, we also see that individuals cluster together based on their population, but also disease dynamic. So epizootic dynamics are in red, and enzootic dynamics are in black. And I wanted to try and identify what might be driving these differences. So initially, I just looked at the composition of the most abundant phyla on their skin, and indeed, we do see some differences. So the 3 populations to the left on this graph are the enzootic populations, and we see that they have a higher relative abundance of Actinobacteria, Verrucomicrobia, and planktomycetes, which are much lower in the epizootic. And interestingly, in the epizootic populations, the most susceptible population, which is RLA, we see an increased relative abundance of chlamydiae. Now, chlamydiae are obligate intracellular pathogens, and they tend to infect mucosal epithelial cells. So the frog's skin, one would imagine, would be an ideal surface for infection to occur. And we do see this high abundance in RLA relative to the other populations. Interestingly, there have actually been some previous studies that have shown that infection with members of the chlamydiae does actually cause mortality. And indeed, one study found that a co-infection of BD and chlamydia in some African dwarf frogs showed over 90% mortality. So there is some evidence to suggest that perhaps, at least in one population, the more severe declines could be due to a co-infection. To look more specifically at individual taxa or groups of taxa that might be driving the differences in the bacterial communities, I did an indicator analysis which essentially identifies taxa that have a high abundance and are highly specific to a predefined group. So in this case, enzootic or epizootic dynamics. And the first point I make is that we're actually finding a conserved set of bacteria that are commonly associated with the disease dynamics in the populations that show them. And this might suggest that there is a common set of selection pressures associated with disease dynamic that means that certain taxa are enhanced. Looking at the composition here, one interesting finding is that we actually seem to get the actinomycetales coming out was quite common. And there's been a lot of work on this group because they're really functionally important, with about 50% of secondary metabolites of known antibiotic function thought to be derived from them. The other thing I would say that's really interesting is that a lot of the OTUs, the bacteria that have been identified in both of these groups, have actually been tested in the lab. And what we find is that there's actually a huge functional variation in terms of BD inhibition and BD enhancement. And that really just highlights the limitations of doing these kind of taxonomy-based approaches, because we can't really gain any information about function. So, moving on, just to summarize, what we've shown is that there's reduced bacterial diversity and significant differences in community structure associated with disease dynamics, and that there seems to be common selection pressure, which means that certain bacteria do well under certain disease dynamics, and understanding what that selection pressure is will be really interesting for future work. The next steps for me, so we have these interesting patterns but no function. So what I've actually done last year was to take mucosal metabolome from these animals in the field, with the main aim of trying to identify whether compositional differences in the microbiome are actually associated with differences in the metabolome, and therefore might actually have a functional effect. We're also pairing this with controlled laboratory experiments, so we can see how infection is changing the microbiome structure and metabolome in the lab. And I think the main point is that in a lot of the literature, people have been focusing on isolating individual taxa from frogs and then looking at their in vitro functionality against BD. But this data really highlights that it's more likely to be communities that are being selected for. So we should move towards that kind of analysis. I'd just like to thank my PhD supervisors for their support, and my lab help and field support. Thanks.
It's interesting, actually, that Thorin should mention at the start of her talk that I left uni wanting to work with birds. And it turns out, actually, I thought the best way to do that would be to don a wetsuit and hug a swan, and may explain why I've now moved out of that into kind of the microbial world. So we have plenty of time for questions.
Did you identify this as the hypervirulent GPL strain of BD? And do you think there might be reason to believe that the differences in virulence between BD strains might actually be caused by how they interact with the microbiome, without there necessarily being any mechanistic differences with how they directly interact with the frogs or amphibians' physiology?
This was all done with GPLs. I think there could be an interaction between the pathogen and the different isolates and the microbiome, but we haven't tested that, and that's something we'd actually like to look at in the future. Rachael Antwis has actually tested differences in inhibition of bacteria with different GPLs, and shown that there is a difference based on the GPL, which is really interesting.
Yeah, this is a somewhat similar question, but it wasn't quite clear from what you said whether you assume that the lower diversity in the epidemic areas is actually pre-existing, so the epidemic goes off, or whether it's a result of the epidemic.
Yeah, so because we've only got one, in terms of the microbiome, there's only one time point, we can't test that. That's why I'm doing the laboratory challenge experiments, to see if there is a negative effect on bacterial diversity when you infect an animal with BD. So that should help to clear up that question. But the answer is we don't know what's driving that specifically right now. There's one down there.
So I appreciate the interaction between different members of the microbiota, bacteria and fungi. So you have a fungal pathogen. Are you considering also to investigate the fungal microbiota of those animals to see whether you may have interesting patterns?
Absolutely, yeah. So I did this sampling in 2015, but I went back this year and essentially did the same sampling scheme, but included metabolome. So we'll have some kind of functional information, and we'll also include the fungal analysis on that as well. So it'll be a lot more comprehensive.
Okay, correct me if I'm wrong here, but I think you're assuming that infection is causing the changes in the bacterial community on the skin. Is that right?
Not necessarily. That's something that we need to test. The point I'm making is at the moment we have a pattern. So that's why I want to pair it with the laboratory challenge experiments, because it could just be that those populations naturally have a lower diversity for, just general ecological reasons. If we had a time series of the bacterial communities since the initial onset of infection, that would be really interesting. But we don't, because this was the early 2000s, and the focus of our lab wasn't kind of community ecology of the microbiome. But the lab experiments should help with that.
Yeah. I was just wondering if you had any populations of the same frog where there wasn't any BD, where you might be able to check their skin communities to see if there's a big difference.
There are, but they're a lot further away in the Pyrenees, so they're several hours. I have actually done that, but I can't remember. Yeah, I did. I actually included in this sampling, I think, a naive population, and we did find that clustered differently as well. So general infection has an effect, is the bottom line.
Right, I think we should move on in the interest of time, so let's thank Kieran once again. And now let me introduce our final speaker from the session, who is Dr. Kayla King, who is going to talk about the drivers of microbe-mediated protection in hosts and the consequences of that protection.
Great, good morning. So virtually all plants and animals, much like all of you sitting here in the audience, are covered in microbes. You know this. But this realization has fueled unprecedented interest in the multifarious effects of microbes on the biology of their hosts. Now this is because, as you've been hearing, microbes need not be merely passengers or even parasites. In fact, they can provide their hosts with a great many benefits. In fact, evidence is mounting suggesting that they can provide their host with benefits by reducing their susceptibility to enemy attack, enemies from parasitoids, pathogens, and parasites. And the micromediated protection does indeed seem to be a widespread phenomenon. As you've been hearing, it might be occurring on frogs, on insects, solitary and social, on vertebrates, including humans, and of course also in plants at the root-soil interface and on leaves. Also, these protective traits might make these microbes attractive candidates for disease control. So protective microbes in the form of bacterial viral phages are being applied as phage therapy. We have probiotics being used. I'm sure you've all heard of fecal transplants. That creates a nice mental image. But we have the wholesale transfer of microbiomes from the healthy to the sick, all to treat very bad human infectious diseases. Moreover, bacteria such as Wolbachia is being piloted to control the spread of very bad vector-borne diseases from Zika to dengue. And also, mycorrhizal fungi can be protective microbes and protect agricultural plants from infectious diseases. So protective microbes are relevant, and they seem to be widespread. The more we look, the more we find them. However, our understanding of the evolutionary processes and indeed the ecological processes underlying this micromediated protection really remain elusive. Now we can think of some ecological mechanisms by which this protection might occur. So you can imagine you have your microbiome in your gut, and you've eaten something that wasn't quite good for you, food poisoning is imminent, okay? So you have a resident bacterial community, and bad pathogenic microbes are coming in. This protection could result from host immune mediation. So your resident microbes are upregulating the host immune response. And so any parasite coming in really is going to be met with an already primed host. The host is ready to go, ready to fight. Prior niche occupation. Perhaps the microbes within you are so numerous and diverse, they're already occupying all available niches, taking up all the resources, and any parasites, again, coming in, have nowhere to go, have no resources to take. And thirdly, direct interference. So the microbes resident on you are producing some sort of toxin, or they're very good resource competitors, and they're directly competing, directly antagonizing the incoming parasite. So that's great. There are a wide variety of mechanisms which could be taking place, but microbes have great evolutionary potential. They have large population sizes and very, very short generation times. So is it possible that micro-mediated protection and all these mechanisms are indeed the result of selection, contemporary selection, acting on your resident microbial populations? And then secondarily, can microbial parasites evolve to fight back against this line of defense? So firstly, can microbes evolve to protect their host? The data I'm going to show you is a result of a paper that was published earlier this year with very many colleagues here at the University of Liverpool, including Greg Hurst. So the system that we've been using to answer the previous question is that of C. elegans and the various microbes that we can actually shove inside of its gut. So this is a great system for a very tractable system for looking at host-gut microbe interactions, because it has a lifespan of about 4 days from egg to egg, depending upon temperature, of course. But it also naturally consumes microbes in its environment, from rotting fruit to compost. We can put in these worms a variety of microbes. I'm going to talk about 2 today. So one is Staphylococcus aureus, another is Enterococcus faecalis. So they're distributed throughout animal microbiomes, from insects all the way through to human breast milk. And if we allow these C. elegans worms to feed on these microbes, they have very different effects on the worms, but they also establish constant population sizes. So these are the effects on the worms. So here we have a bunch of worms on Petri dishes, and the proportion of dead nematodes after 24 hours on the y-axis, and the various bacterial species here. When the worms have been eating E. faecalis for 24 hours, there isn't much of an effect. These microbes are a little bit bad for them. We see a population reduction of about 0.5%, but nevertheless, when the worms are feeding on Staph aureus, this bacteria is very parasitic. It kills most of the host population. Now, the really cool bit is when we put these 2 microbes together. We see this attenuation of virulence, quite dramatic, in fact. And this is what got us interested in the idea that maybe E. faecalis here is suppressing the virulence of this parasite. It's potentially protecting its host, despite being a little bit costly itself. So we wanted to use experimental evolution to explore this relationship a bit further and see if we could get this purple dot right here all the way down to 0. Could within-host interactions between these microbes, between E. faecalis and Staph aureus, actually lead to the evolution of total suppression by these microbes here? So to test this hypothesis, we set up a very simple experiment where we had 2 treatments. The worms here are allowed to eat E. faecalis. There's about 900 worms in the population. They're very genetically homogeneous, and they're not evolving here. And in this treatment, we have the worms eating E. faecalis, and then they're transferred to the parasite. So this is very much like the situation I mentioned previously, where you have a resident microbiota, and then you have a parasite invading. After 24 hours, the microbe Ephicalis was passaged from dead worms and transferred to the next generation where they met with more isogenic worms. So here what we have is we just have Enterococcus evolving in both these treatments. The resident microbe is evolving here and here, but here it's evolving with a non-evolving host, and here it's evolving against a host and a parasite. So here we have a 3-species community and a 2-species community. And this went on for 15 host generations and was replicated 6 times. So do we see the evolution of enhanced microbe-mediated protection when this parasite is about? Indeed we do. So we very much see that here if we look at the proportion of dead nematodes on the y-axis, and on the x-axis here we have time, we go from this intermediate level of virulence that the parasite exhibits when that other microbe, Ephicalis, is present, all the way down to almost 0. So we see this enhanced protection occur very, very rapidly after 5 generations, and the relationship is strengthened and is also maintained quite consistent for another 10. So again, this is when the parasite is present. We don't see the evolution of enhanced protective properties in the treatment, in the single evolution treatment, where staph was not present at all. Now here's a little interesting quirk. When we take the parasite away, when we take staph away, we see that in both treatments, the virulence of this resident microbe increased a wee bit. So this is a really cool example of a microbe that's crossed the parasite mutualist continuum. When it's on its own in the host, it's a little bit harmful. But when it's presented with a much, much more virulent competitor, the benefits of having it are quite great. It's very much a condition-dependent mutualist that we've evolved in novo. So what happens when we look at the fitness of this resident microbe and this parasite within the worms? Here we have the number of colony-forming units on the y-axis. The blue bars represent the fitness of our resident microbe Ephicalis, and the red bars of our parasite staph. If we compare the blue bars here, we can see that experimental evolution has very much favored the persistence and the proliferation of this resident protective microbe. So we see an increase in the blue bars here. And it also suggests a mechanism by which protection is occurring. I won't get into the details of the mechanism, but what you can see here is that when staph is combined and allowed to co-colonize with a protective microbe, it has a greatly reduced fitness within hosts. So then we wanted to see how broad spectrum our protection was. Is it the case that we managed to evolve a bacteria to protect against a particular strain of staph that we happen to have in the lab, or is there something more important going on here? So we exposed our worms to a range of different parasite isolates collected from a diverse source of infections and from all over the world. We even included 4 strains of the superbug MRSA here. So we exposed our worms to one of the 7 different isolates of staph. We preconditioned them with the ancestral resident microbe, Ephicalis, which was sort of mildly protective, and then the very strongly protective Ephicalis here. And what did we find? This is one of the nicest graphs I will ever produce in my career. I'm quite confident of that. Right, so we very much found that the protective microbe we evolved in the lab was generally protective, even against 4 strains of the superbug, MRSA. And this is quite cool because these worms should be dying en masse. If you look at the red dots here, across our staph strains, the death rates range from about 30% all the way to 60%. This is the strain of staph we used in our experiment. But if they're preconditioned with this evolved protective microbe, they're happily crawling about on a plate, consuming a parasite which should decimate the population. And that's what we see here. So when they're preconditioned with the ancestral resident microbe, we see an intermediate level of death, but it's when they have this evolved protective microbe that things are pretty smooth. And they survive at a high rate. Okay, so we've seen here that our protective microbe can reduce the fitness of the parasite and indeed reduce the negative consequences of infection. So we wanted to know what the longer-term evolutionary consequences of this would be for parasites. Because, as many of you here I'm sure know, it's been well established that parasites can adapt quite rapidly to challenges to their establishment, with follow-on consequences for the severity of the disease that they cause. So the next results I'm going to show you were just published in Nature Communications last month. So can protection drive parasite evolution? So before I go on to show you how we went about answering this question, I need to display for you a quirk in the system. So here we have the number of CFUs per nematode, and this is for Staph aureus. This is for our parasite. Now, when the worms are infected with just this parasite, we have this level of parasite fitness. And when they're co-colonized with the protective microbe and the parasite, we have a reduction in fitness. I've shown you this before already. But the quirk is this. Here is the fitness of our protective microbe. When the protective microbe is on its own in the worm, we have this level of fitness. When it's co-colonizing with the parasite, it does better. It's a bit of a hyperparasite, a facultative hyperparasite. So indeed, we see an antagonism emerging within the microbial community in these nematode hosts. So this is the relationship that we then wanted to explore a bit using, again, experimental evolution. In this case, experimental coevolution. And then to look at the effects on parasite virulence and the evolutionary trajectories that resulted. So the setup was very similar to, as I've already explained for the previous part of the talk, where here we have a worm population of about 900 genetically homogeneous worms. They're not evolving, eating up the parasite. After 24 hours, the parasite was extracted from dead hosts and then passage to the next generation. Here, both the protective microbe and the parasite were co-passaged together. We assumed that they would evolve on similar timescales, even within hosts that are long-lived, for instance, in nature. So this would facilitate potential coevolutionary interactions. And this experiment went on for 10 worm generations and was replicated 5 times. So again, here we just have parasite evolution. Here we have coevolution between the protective microbe and the parasite. There we go. And what we saw is that the parasite that was co-evolving with these protective microbes evolved to be less virulent over time. Indeed, quite, I think, a third less virulent. And we didn't see this effect in the parasite that was merely evolving in a static host population. So this is quite interesting. And in line with the common assumption that virulence is linked within host replication and host exploitation, we very much found that the growth rate of the parasite was related to its virulence. So here we have the percent dead nematodes on the y-axis and the within host growth rate of staph. What we can see is relative to the ancestor here, which is the red dot, our evolved populations clump here, and our co-evolved populations clump here. So whilst co-evolving with this protective microbe, the parasite evolved to become less virulent and have a lower growth rate. That doesn't sound great, does it? However, it indeed adapted to these microbe-protected hosts. So if we look at the number of colony-forming units per nematode of our parasite, again, our parasite fitness, we can see that it is the co-evolved staph once placed in a microbe-protected host, a host with this protective E. faecalis, that does the best. It has the highest fitness. So again, despite being less virulent and having lower growth rate, it performs the best in a microprotected host. It's adapted, which could have some effects for its transmission potential, surely. Right. So we then wanted to explore the mechanism underlying this reduction in virulence and reduction in growth rate, because were these traits as a result of this antagonism with the protective microbe in the microbiome of these worms? So from a look in the literature, a very well-studied molecule that relates to both the growth rate and the virulence of a suite of bacterial pathogens, including Staph aureus, is siderophores. So siderophores are iron-scavenging molecules which are beneficial for bacterial growth. They essentially allow bacteria to chelate the iron in the environment, particularly in environments that are iron limited, probably host environments, and then bring it in and use it. And the more siderophores that a bacterial pathogen produces, the more resources it's able to extract from a host, and the better it grows. Interestingly, though, they're a public good. So they're liberated into the environment once produced by a bacterial pathogen, and anything else can use it, even other bacterial species. So I'll just ask you to sort of cast your mind back at the little quirk I mentioned previously, where our protective microbe was actually benefiting from its interactions with the Staph aureus parasite. So this became very much a candidate virulence mechanism for us, and a candidate mechanism by which ecological interactions between our protective microbe and our parasite occur. Right. So I had a very clever PhD student set about measuring siderophore production. And indeed, she found that relative to the ancestral strain of Staph aureus and the evolved lineages, the co-evolved populations indeed produced fewer siderophores. And siderophore production was positively correlated with virulence and with growth. Okay, so siderophores became even more of a mechanism that we were targeting. However, we had yet to conduct the confirmation of causality, and to be able to directly link it with the antagonism between this protective component of the microbiome. So again, just a reminder of this antagonism, where our protective microbe Ephecalis benefits from its interactions with staph. Is it benefiting from these siderophores, this public good? So I'll go through this graph in steps. So here we have, outside the host, this is in vitro culture, the number of colony-forming units per mil of our resident protective microbe, Ephecalis. Now here is the end point of growth of the protective microbe on its own, this line here. And then we co-cultured Ephecalis with a variety of different parasite lineages, with the ancestral one, with the evolved parasite lineages, and with the co-evolved parasite lineages. Now, here is that benefit, the difference between this red bar here and the line. So here is where Enterococcus, again, is benefiting from interacting with the parasite. This benefit is lost in the co-evolved parasites. The co-evolved parasite isn't benefiting the protective microbe as it once was. Once we added more iron to the environment, we saw that this benefit was restored. So this, to us, was very much evidence of a confirmation of causality, of iron being the resource that these organisms were competing over, and siderophores being at the interface of that antagonistic interaction. Right, so in conclusion, the presence of an evolving protective microbiome or microbe can fundamentally alter our understanding of the benefits that the microbiome can give their host, and indeed of host-parasite interactions. What is host resistance? What barriers do parasites have to overcome to establish? So I hope I've shown you that we've established that microbes can be a costly yet evolutionarily dynamic arm of host defense. We had hypothesized that because of their great evolutionary potential, again, large population size, really rapid generation times, that these microbes could evolve to form a key component of host defense. And indeed, we saw the emergence of this benefit from direct antagonistic interactions between the protective microbe and the parasite. This protective effect was powerful and indeed general and rapid. So within 5 host generations, which is essentially the equivalent of 5 days exposure to this protective microbe, did we see the evolution of these traits? They're powerful. So staph should be killing most of the host population, but if these hosts have this evolving micromediated defense, then almost none of them perish. And this effect was general. So we saw suppression against a wide range of really, really bad parasites, including 4 different superbugs collected from around the world. But of course, we must remember that parasites can evolve back. They can evolve in response. They can fight back. And that's what we saw. So parasites can evolve in response to the microbiome, and this might have the effect of shaping disease severity both in nature and in clinical situations. Thank you.
Thank you, Kayla, for a great talk, some really elegant experiments with some really fascinating results. So we've got plenty of time for questions. See one at the back.
The other bacteria in here, particularly food, I mean, the Staph aureus and the Epicales, that's not the food source, is it?
No. So we feed them to E. coli before they're exposed to E. faecalis. But the E. coli does not remain resident in the worm at the same time that these bacteria are. We've confirmed that.
Okay, I mean, if I may have a second question. What I was wondering was the work by Heinrich Schulenberg that's looked at the microbiota of C. elegans, and so it seems to be a standard or a resident one there. And I wondered actually how this, what you described, would behave in the presence of that microbiota.
In a natural microbiota?
Yeah.
Well, I'm hoping the results of some timely grants come through. So I can answer that exact question. We would like to know if the protective effect remains when there's a greater diversity of microbes present in the worm. And then secondarily, how does the presence of a protective microbe, indeed early in life, from treatment, from the environment, how does that then shape the subsequent structure of the microbiome? Yeah, great.
Well, this question more out of curiosity, but let's see. You got a frighteningly fast effect of protection. Right? In a few generations, if the E. coli, the protective microbiome is evolving, protective effect is huge and it's against a large variety of other bugs. So one wonders why the host is so neutral if there's no selection by the Staph aureus in the controlled treatment. You evolve the community of the bacteria, but it seems that the host does nothing to, let's say, retain the good protectors, so to speak. And that's a bit surprising in a way, isn't it?
Thank you for asking that question. It's almost like I planted you there. So indeed in these experiments, we did not allow the host to evolve. However, we have results now suggesting that when we do allow the host to co-evolve, it indeed takes advantage of this protective microbe over evolutionary time and either evolves to resist it less or to actively farm it, which is kind of cool, which is like a de novo mutualism, right? So we're following those results up now. Yeah, good insight.
Okay, this may sound a bit naive because definitely worm biology is not.
No worries. Go for it.
My question is, how do you square host immunity in your picture? So in particular, for instance, so if you treat your protective agents, A. faecalis, you autoclave it, and then you shuffle inside your worm, do you see this protective effect again, or the protective effect declines in a way that it requires a living organism, basically?
So when we take E. faecalis out of the host and maintain it for a while, and put it back in. No,
maintain it for a while in an autoclave. So you eat inactivate, you kill it, and then you put it back. Have you considered?
No. So I didn't actually show the results. We have a good understanding of the mechanism of protection. So we know that the microbes are evolving to produce more superoxides, which suppresses the parasite both in vitro and inside the worm. I didn't show those data, but and dead E. faecalis don't produce them.
Okay, folks, I would just very much like to thank Kayla and indeed all of the speakers in our session for a truly wonderful diversity of talks. And on behalf of Rachael, Antwis and myself, thank you very much for coming.